Hi guys, and welcome to Biological Sciences for Year 10. This is Activity 8 where we're going to be looking at codominance. Now in the previous activity we looked at the concept of incomplete dominance, where we had two alleles which came together and basically produced a mixture. In codominance what happens is there is no mixturing, there's no mixing of the alleles to get a different trait. What happens is the alleles are equally dominant, and as a result their features actually come through in the individual. So basically we'll find that codominance is where an offspring shows both of the parents' characteristics. Now one of the best examples for this is the blood grouping system. Now in the blood grouping system we've got four main types of groups. You can either be, your blood group could be an A group, it could be a B group, it could be AB, or it could be an O group. Now these are controlled by a variety of different alleles. Now these alleles basically are written with a capital I. Now it's a capital I with a superscript of the type of blood group above it. So you could be capital I A, capital I B, or little I, which is the recessive form of the of that blood grouping. Now the I basically comes to the protein which is necessary to produce the blood grouping system itself. Now basically what we're going to do is very quickly run through how codominance comes about, and hopefully you'll see through the example how you can see that the two alleles basically have equal strengths. So if you look at this table below, I'm showing you the genotypes of the main blood groups. So in blood group A, remember you're always going to have two alleles. Now if you're blood group A, you can basically have the allele IA, which is a homozygous dominant genotype, or you can have the heterozygous form, which is IA and then the recessive I. Now the recessive I basically just does as it's told, and the capital IA basically dominates over that recessive form, so you're still blood group A. Now the same is true for B. You can have the homozygous IB, or you can have the heterozygous IB little I. Again, the IB would dominate over that recessive form. Now think about what happens when you've got two parents. One is A and one is, for example, B, and they happen to be homozygous. Now if they're homozygous, that means that's IA joined with IB. Now your genetic crossing should be at a level now where you're probably thinking there going, well, what's going to happen is you're going to get all of your offspring which are going to be IA, IB. Now in this case what happens is both the IA and the IB have equal dominance, so their characteristics are shown through in the parent. Now it's not a mixture, and as a result, because it's not a mixture, we basically call them AB, because they're showing A traits, they're also showing B traits. So there's no mixing whatsoever, so they just exist as IB or IA. So as a result, the blood group is AB. Now finally, what you can then have on this blood grouping is if you had two recessives come together, then you would be blood group O. And you can see the percentage of Australians on the right hand side, which basically shows that we've got 38% in blood group A, 10% in blood group B, 3% are AB, and 49% are O. Now what that then means is that you can only give certain blood groups to specific people. So if you're a blood group A, then you can only be given to blood group A. If you're B, you can only be given to B, or you can be given A and B. So the result is that you can basically determine what's going to happen with your genotypes, but the key thing that I want you to understand in this case is when we have a form of codominance, we've got equal dominance for both of them, but their traits don't mix together, they stay on their own. So I hope that makes sense to you. Again, it's more of a case of just having to understand that that's the way it is, and once you've learned what the word codominance means, you then just apply it to your genetic cross. So in a question you may be asked, this is an example of codominance, show what comes through. You would then basically state that the phenotype has to be one of each. They're together. You can't have that mixing. Okay, so I've put together some more exercises in activity 8B. Have a go at those, see how you get on, and yeah, join me again. Thank you for watching.